So welcome everyone to our first science seminar of 2024. And I'm not going to introduce our speaker today. Dr. Masumi Majumdar is going to introduce our speaker today, but I'll just let you know that next week we have another online talk, but hopefully we'll be in person back in 447 in the Brody building. Even though our speaker will be on Zoom. But this was kind of a special case of sickness and needing to be at home and various things going on. But Dr. Temitope will be talking to us next week about quasi-groups, loops, and some of their applications. So it'll be a mathematics talk next week, I believe. So, Dr. Masumi, I'm going to let you introduce Dr. Allan, and then we'll have our talk, and then we'll have time for some questions at the end. Okay, go ahead, Dr. Masumi.
Thank you, everyone. Thank you, Chris. And good afternoon, those who do not know me. I'm a faculty member at the Department of Biology at Brandon University and Tier 2 Canada Research Chair in genotoxicology. So this is honestly my honor to introduce to you Professor Alison Allan. She is the Canadian leader in the research area called circulating tumor cell. And also she's at the front side of the technology development. What it does, it helps you to detect, use biofluidics or using these tumor cells, you can detect cancer in biospecimens. So she's our Canadian leader in that. So she's also actively involved in many prognostic and predictive blood biomarker for cancer detection in local, national, and international multi-center clinical trial. She's part of that. She's a professor at the departments of oncology and also and chair of anatomy and cell biology at the Schulich School of Medicine and Dentistry at University of Western Ontario or Western University. She's holding a scientist position at the London Regional Cancer Program, and she's also the scientific director of LRCP Biospecimen Lab. There's lots of portfolio, I can go on and on, okay? She earned her BSc and PhD degree from the University of Guelph on molecular bioscience and also molecular biogenetics and medicine. She did her postdoc at Western University, where I did few years later my postdoc as well. It seems like that she earned those degrees just to get a job at somewhere, because her CV shows me that she is still taking training, every possible leadership, mentorship, and EDI inclusion training. It actually took me a few hours to summarize and just cherry pick from her battery of recognition and award. These are very impressive awards she received. She received Dean's Award of Excellence for Translational Research. Hold on, and also for all around Scholar Excellence Award. She actually started her position at Western with the CIHR New Investigator Award, and she also received Early Researcher Award from Ontario Ministry of Research and Innovation, and many other. I can go on and on. Just to tell you that her trainees award and recognition, it is actually taking 3 pages in her resume. Okay. And for students here, I'm not sure if Dr. Allan ever paid for her tuition or anything. She received all graduate and postdoctoral scholarships and fellowship Ontario could offer her. So she has all of it on her, on her portfolio. She is serving. This is how her expertise shows on. She's serving Tri-Council, CIHR, NSERC Discovery Grant Panel for 10 years, CFI, CRC, and also internationally Wellcome Trust, Cancer Research UK, NIH, and so on. And she's serving all reputed, for instance, top tier publisher, Nature, FASIB, Wiley, PNS, numbers of journal. She is serving as the scientific reviewer. Just a little bit about her, how I know her. She is an excellent mentor, and she runs many youth outreach activities. For instance, Let's Talk Science, mentorship workshops, her mentorship, and many even at the AACR, or our reputed cancer research organization. She is serving as a either the celebrity judge or a faculty mentor. Her mentorship skills have a strong footprint, which includes numbers of master student, PhD student, and postdoc. But her level of traineeship goes from high school level to the junior faculty level. So she is mentor of that many level of, trainees. On a personal note, first time I met Alison Howe when I was attending. I just started as a postdoc at Western, so I was at a conference where back to back, every award going to two students, one PhD, one master student receiving all award presentation, and I was looking for the mentor or their supervisor because she's working on breast cancer, kind of a very new field for me then. And I find someone very quiet and very shy, but guarded by at least 10 different students and professors talking to her, and seeking all advice. So very strong leadership quality. She inspired in many occasions, many way. I would say that few years ago when I was going through really struggling, at some milestone to reach, I always seek her advice, especially if we need some expertise and knowledge in breast cancer research, because biomarker research is very new to me as a faculty member when I started, and she was the leader, already established one. So when someone is so busy with so many admin work, she has very strong funding support from CIHR, Canadian Cancer Society, and she had grant from US Department of Defense, LRCP, also Prostate Cancer Canada. And she is at least part of 10 different clinical trials. On publication front, she published over 75 peer-reviewed scientific articles and 12 book chapters. On citation scale. Something I find in her resume, new that to me, is I-100 index. What does that mean? It means that how many of your publications are cited over 100 times. And for her, it's 20. So that just tells you her leadership and the quality of the research she is doing, and the mentorship and legacy she is, developed as a faculty and as a professor. So this is my honor. We are here to learn a lot about that seed and soil hypothesis, which I often include in my classroom when I'm talking about cancer stem cell. I took few of your essential hours as maybe minutes, but I couldn't stop myself because I have seen you as a woman in STEM, how you balance work and life as a young mother when you started. So all the flashback came in front of me, running from one campus to the other, all things coming back. So I really warmly welcome you to BU, and please share your research with us.
Oh, thank you so much, Dr. Majumber, for a great introduction. That sets the bar pretty high for me, telling a great story. So thank you for that. So I will jump right in. So I'd really like to thank everyone at Brandon for this introduction. It's my pleasure to be here today. And I'm coming to you, as you heard, from Western University, which is located on the traditional lands of the Anishinaabe, Haudenosaunee, Lenape, and Chinungtunk nations, on lands connected with the London Township and Sombre Treaties of 1796 and the Dish with One Spoon Covenant Wampum. So just to start, in terms of kind of giving you a framework, you heard a lot about my research, but we We like to, so when I started the lab 18 years ago as a new faculty member, I used to say we were a breast cancer lab, but really we are a metastasis lab. And so our overarching focus is on understanding cellular and molecular mechanisms of cancer metastasis. And our research goals are really broken up into two main areas or two main projects. And the first is to gain a greater understanding of the biology of cancer metastasis, which is the spread of cancer away from the primary site to different organs like the lung and the brain. And also for us to use biology to investigate opportunities for development of new therapeutic strategies. And then the second area that you heard about, which is more kind of clinical, translational, is that we do a lot of work developing and validating clinical blood biomarkers for monitoring metastatic progression and therapy response. And as a basic scientist, I have the privilege of collaborating with clinicians on clinical trials, which means that my work can move more quickly into the clinic to help patients. But today we're going to talk about biology because that's really my first love. And I know this is a mixed audience. And so I want to try to, rather than kind of, dig really deep into nitty gritty science that we do, is to more give you a story about the journey that we've been on over the last 18 years since I started my lab in terms of investigating cancer metastasis. And so just to set the framework, so those of you who have a background in cancer biology will recognize this diagram and this term, the hallmarks of cancer, which was coined by two very famous cancer biologists, Douglas Hanahan and Robert Weinberg. And they basically took decades of, or a century probably, of cancer research and broke it down into these very specific hallmarks of cancer that every cancer needs to do in order to be successful in terms of cancer disease. And so there's kind of the normal things that you might expect from cancer, like sustaining proliferative signaling, evading growth repressors, resisting cell death, enabling immortality. And then over the years, as our knowledge has built in the field, there's some other really more complex but also super interesting hallmarks, which includes cancer cells regulate cellular metabolism. They do a great job at avoiding immune destruction. I'm just going to switch my pointer here. There's an emerging role of tumor promoting inflammation in the microenvironment. Cancer cells need to be able to get into the bloodstream in order to spread to those distant sites. Cancer cells are often very genomically unstable, which allows mutations to accumulate so that they can start to do all these hallmarks. And of course, what we're going to talk about today, which is the focus of our work, is this important hallmark, and in many cases, sort of the later stages of cancer, which is activating invasion and metastasis. So the motivation for our work, again, is driven by the clinical need. So today we're going to talk about breast cancer specifically. And this past year, there was almost 30,000 new cases of breast cancer diagnosed and 5,500 deaths from breast cancer. And so breast cancer is the leading cause of cancer diagnosis in Canadian women, and the second leading cause of death. The first leading cause is still lung cancer. And breast cancer is arguably one of the sort of better outcome cancers, but almost all, like more than 90% of breast cancer deaths are due to this process of metastasis. And one of the reasons for this is that most therapies are non-curative in the metastatic setting. So researchers really were motivated by the need to understand the basic biology of metastasis, to develop new drugs and treatments based on this biology, and also importantly, trying to find strategies to prevent metastasis. So even though metastasis is a very deadly process, it's actually quite inefficient. So this diagram kind of outlines the steps that a cancer cell needs to go through in the metastatic process. So if this area right here at the top is the primary tumor. The first thing the cancer cell has to do is get into the bloodstream or the lymphatic system in order to escape from the primary tumor. And if the cancer cell goes into the lymphatic system, it does eventually need to get into the bloodstream to go to very distant sites. So once it's in the vasculature, so it does this process called intravasation, which is basically invasion from the primary site into the bloodstream. The cancer cell has to survive in the circulation, and that's survival from immune attack, but also from just things like shear stress from the pressure, physical pressure of the blood flow. Then a cancer cell will come to a secondary site, such as the lung, where it will usually arrest in the capillary bed, but it has to then extravasate or invade out into that secondary site. And at the secondary site, the tumor cell has to then not just survive as a single cell, but then initiate growth into a bigger metastases. And then it also needs to maintain growth in that secondary site. And eventually a metastasis, if left untreated, will grow to the point where it can also start the whole process again, going back into the bloodstream and seeding secondary metastases. So if we look at the efficiency of this whole process, we know from lots of really exciting work in the late 90s by my colleague, Dr. Ann Chambers, that about 80% of cancer cells will actually get all the way to the secondary site and into that secondary site. But where the inefficiency comes in is that only about 2% of these cells will be able to actually initiate growth in this foreign microenvironment. And less than 0.02% will actually persist. So this really, from our perspective, we're really interested in what makes this very small subpopulation successful. What are their characteristics? And so just as I was starting my lab in 2006, there was this emergence of the cancer stem cell hypothesis, which I'll explain in a minute. But it made us think about whether or not this very small population that's able to metastasize are actually stem-like cancer cells. And so, as I said early, when I was just kind of later in my postdoc, just starting to think about being a new PI, there was several papers that were published in breast cancer. Breast cancer was the first solid tumor to identify so-called cancer stem cells or tumor-initiating cells. And what was observed that cancer cells with certain markers, which again I'll talk about in the next slide, which includes cell surface expression of a receptor called CD44 and negative expression of another receptor called CD24, as well as activity of an enzyme called aldehyde dehydrogenase. This phenotype, the cell phenotype, identifies stem-like cancer cells or tumor-initiating cells. And when you inject these into a mouse, they always form a primary tumor, even at very, very low numbers, as low as 10 or 20 cells. If you take the rest of the population that don't have this cellular phenotype and you inject them into the mice, even at very high numbers of cells, like 1 million cells, they don't form a tumor. But at that time in the field, everyone was really focused on, do these cells form a tumor? But in breast cancer, our surgeons and our radiation oncologists are really good at actually treating and in many cases curing breast cancer as long as it stays confined to the breast. And so we are more interested, because we're metastasis researchers, whether these cells are also contributing to metastasis. So again, for the grad students, like, it's always great to go and back and look at the literature, and we observed that the classical properties of normal stem cells are very similar to observed experimental and clinical behavior of metastatic breast cancer cells. So both of them have high replicative potential, which includes both self-renewal and the ability to give rise to different cell types or heterogeneous population of cells. Both require a supportive microenvironment, or in the stem cell field, it's called a niche, in order to grow. And both have an increased capacity for self-protection against harsh environments, toxins, and drugs. And this is really important, obviously, in cancer cells, because it means they can develop resistance to chemotherapy and radiation. And so we hypothesized that the subset of cells within the primary breast tumor that are capable of reinitiating growth to form these metastases in distant sites may in fact be tumor cells that have retained or reacquired these stem-like properties. So what are these markers? So as a cell biologist, the good news is that these are not just, markers that allow us to use technology to pull them out, but they're actually they have an important biological function. So if we look at the aldehyde dehydrogenase, this is an enzyme that converts retinol into retinoic acid. It's very important in terms of normal cellular differentiation during development and in stem cell self-protection. And then again, from a functional cell biology perspective, it's actually a functional marker, so it fluoresces green inside the cell only when the enzyme is active. So it helps us isolate the cells through fluorescence-activated cell sorting. And then the combination of this ALDH with the high expression of this CD44 receptor. CD44 also has known functional importance in cancers. So it's a receptor for osteopontin and hyaluronin as canonical ligands. And also it has a number of non-canonical ligands which will become important later in the talk. And CD44 is really important for cell-cell and cell matrix interaction, so the ability of cells to talk to each other. And it has known functions in migration and adhesion, cell migration and cell adhesion during metastasis. So the first question that we had was whether these human breast cancer cells with this ALDH high CD44 positive phenotype play a key functional role in breast cancer metastasis. So my very first PhD student, Alicia Croker, who's was super smart and super creative and actually is now a director of patient engagement at Health Canada. Her project, she took 7 different human breast cancer cell lines with varying metastatic ability. And then she analyzed their ALDH and CD44 expression. We used that to sort the cells using fluorescence activated cell sorting. So we ended up with 2 populations of the ALDH high CD44, which are our cells of interest, or the rest of the cells, which were ALDH low and CD44 negative. So we isolated them, and then Alicia did a whole battery of functional tests, including in vitro tests like cell growth and colony forming ability, things like migration and invasion, which are an in vitro, a way to look at some aspects of metastasis in vitro. And then, of course, we need to study, because metastasis is a whole body or systemic process, we studied their ability to form tumors and metastasize in vivo in animal models, in mouse models. So what Alicia observed was that ALDH high CD44 positive breast cancer cells do demonstrate enhanced metastatic behavior across all of the assays that we looked at. So we looked at in vitro, we saw significantly enhanced migration, invasion, and adhesion, and then really significant increases in lung metastasis. And so this is just a graphical representation, but here's what the lungs look like at necropsy. So you can see in the stem cell population, we see lung mets, these little white aspects, those are all lung metastases. And then in the non-stem cell population, we see a set of pretty clean lungs. And then this is just what it looks like histologically. So this is what a normal lung, this is in the mice that were injected with the non-stem cell, and you can see a nice healthy lung, and you see in the stem cell population that we saw the lungs filling up with metastases. And so, as PIs tend to do, to summarize the blood, sweat, and tears of a PhD student, 5 or 6 year thesis in one slide, this is what Alicia found, is that, that was an example of the MDA-MB231 cells, but she repeated this in all of these different cell lines, and the pattern that we saw was that the more tumorigenic and the more metastatic the cell line, the greater the ALDA high CD44 positive content was. But if you, regardless of the aggressiveness of the cell line, what we saw is if we pull these stem-like cells out, we enhance tumorigenicity and metastatic ability. And so subsequent students in the lab, Maurizio and Irene, went on to assist Alicia with a lot of this work, and we observed a number of things. We found, obviously, so we were the first group in the literature to report that these stem-like cells not only are initiating tumors, but they're also metastatic. And Dr. Majumdar mentioned the citations, and this first paper of Alicia's remains one of the highest cited papers of ours, of mine and every other collaborator. Who they're always saying, oh, Alicia's paper got cited again. So we're really thrilled to kind of sort of set the tone in the field. We also observed that these cells contribute to both chemotherapy and radiation resistance, and that, you know, from a functional perspective, this ALDH enzyme, the two specific isoforms, 1A1 and 1A3, also play a mechanistic role in both metastasis and treatment. So our next question was whether, you know, are these cells just super aggressive? Is this a general phenomenon, or is it organ specific? And so I'm just going to pause and kind of give you guys a little more background. This is where we get into the seed and soil part of my title. So metastasis is not a random process. So each type of cancer, be it breast cancer, prostate cancer, colon cancer, they have a specific pattern of metastasis. So in breast, so they go to certain organs. So prostate cancer, for example, the majority of prostate cancers go to bone. But in breast cancer, the four main distant sites of metastasis are the lung, the bone, the brain, and the brain and the liver. And the reason that metastasis kills patients, in addition to the therapeutic resistance, is that the breast is not an organ, a critical organ. So a tumor, if you remove the breast doesn't keep you alive. So if you remove the breast, there's obviously cosmetic and emotional aspects. And if you're a young woman, there's some significant implications, you know, for childbearing and breastfeeding. But when the cancer cells spread to critical organs like the lung, they really affect your breathing. If it spreads to the liver, it affects the function of your liver to detoxify. If it spreads to the brain, it impacts neurological function. If it spreads to the bone, it starts to affect your immune function, and you get a lot of fractures, etc. So we know that breast cancer spreads preferentially to these four organs, and there's a few hypotheses about why this is, but the one that has kind of withstood the test of time is called the seed and soil hypothesis, which was proposed by Stephen Paget first in the 1800s. And he was a British surgeon, and he proposed that when a plant goes to seed, its seeds are carried in all directions, but they can only live and grow if they fall on congenial soil. So in this case, the seed is the cancer cell, and it can only survive and proliferate in secondary organs, or the soil that produces growth factors appropriate to that type of cell. So the analogy I always like to use is I have my neighbor at home. He's retired. He's like obsessive about his lawn. He always puts, you know, all of these pesticides on it. And so it's a very inhospitable environment, and he never has any dandelions or any weeds on his lawn. I'm always in the lab, busy doing science stuff. And so my lawn is a very hospitable place, and so it just fills up with dandelions. So that's the kind of idea about the seed and soil. And so then if we come back to this first idea about metastasis being inefficient, what we now believe is that it's really, this inefficiency is driven by a balance between the seed and the soil. And so when we were starting to think about this organ tropism or seed and soil aspects of our studies, There was a series of papers that were published out of a big lab at Memorial Sloan Kettering in New York, Juan Massagué's lab. And this group identified genes that mediate breast cancer metastasis to specific organs, including the lung, the brain, and the bone. And you'll see these are published in big journals, published 2 in Nature, 1 in Cancer Cell. And these were sort of the first real definition of what are the molecular things that are driving the seed and soil hypothesis. But again, they were focused on the seed. So they're focused on the cancer cell. And the way that Massagué's group did this work is so they took this MDA-MB231 breast cancer cell line, and they injected the cells, and these are whole population cells, not sorted or anything, injected them into nude mice, and they allowed into the mammary fat pad, which is the where you would inject it similar to a breast in a human. They allowed these tumors to grow and then also to metastasize. And then when they metastasized to the brain or the bone or the lung, they harvested them from those organs and then they re-injected them back into the mice until they essentially had selected organ-specific or organ-seeking metastatic variants to the brain, the bone, and the lung. They then did a microarray analysis, which was the cutting-edge technology at the time. Now you would use RNA-seq. And what they found is a gene signature for each of these metastasizing sites, and they validated in patients. So for the lung, for example, the lung metastasizing signature, they looked at a cohort of lung patients and their primary tumors, and they saw that they could get a match in the signature, which would predict whether a patient is going to metastasize. So, and we're not going to go into the details here, but here's a list of just some of the genes in each of the signatures, and you'll probably notice there's some overlap in some of them. So this things like matrix metalloproteases, which of course break down and help with that migration and invasion back and forth, and some angiogenesis related genes like angiopoietin and VCAM, but combined, it's this sort of signature that gives the specificity. So how does this work into kind of what our thinking was at the time? So Massagué's work was really exciting. It uncovered particular genes that can mediate the role of the cancer cell or the seed, but investigation of the factors contributed by specific organ sites or the soil has really received much less attention. And the reasons for this, at least the reasons we think, are that it's actually quite technically difficult to study organ microenvironments in a tissue culture dish for obvious reasons, but it's also challenging to identify and study specific molecular factors in detail using in vivo models of metastasis. And also in the context of our cancer stem cell work, how that overlapped with metastatic organ tropism and the seed in the soil hypothesis were quite poorly understood. So then along comes another super bright grad student, Jenny Chu, who decided that in order to address this, she was going to develop, since there wasn't really a great model system, she was just going to develop her own model system. So Jenny harvested the common sites of breast cancer metastases, these organs from mice, from normal mice, and she cultured them in media. And then the organs, so basically she developed conditioned media from each of the organs where they release soluble factors specific to each organ. She spun that down to get rid of the cellular debris. And then she used that to treat breast cancer cells and assess functional behavior, again, things like migration, invasion. And then we started to look at some protein and gene arrays and proteomics. And so before we kind of jumped into, a lot of studies we wanted to, it's always good to validate. If you're developing a new model, you want to validate it against what's in the literature. So we initially just took bone conditioned media and lung conditioned media. We treated breast cancer cells with that, and we saw that they increased several of the genes that were within these defined organ-specific signatures that Massey identified. And then we sort of flipped the experiment around, and we actually obtained the bone-seeking and lung-seeking cells from Massey's lab. He kindly shared them with us. And then we treated these cells with all of the different conditioned medias. And what we observed was that the bone-seeking cells really liked the bone conditioned media, and this is migration. And then the lung-seeking cells really liked the lung conditioned media. So we were relatively confident that the readouts we were going to get from this model system in the experiments we wanted to do should be reflective of what's going on in breast cancer metastasis, or parts of the process at least. So then coming back to the cancer stem cell idea, what we observed, so we went into it without a hypothesis. Well, I shouldn't say that. We actually did have a hypothesis, but it proved to be not true. But we wanted to treat, to separate out these cells. So we sorted out the stem cells versus the non-cancer stem cells. We treated them with the different organ media, and we actually predicted that they would like the bone conditioned media best because there's a defined stem cell niche in bone. But actually, surprisingly, they liked the lung conditioned media the best, which was really interesting. But again, like, really good lesson for grad students. Strange results are not bad. Strange results are just something really interesting to pursue, which is what we did. And we also observed in vivo, when we injected the cells, that even though they did go to some other organs, they always went to the lung. So they had a preference for metastasizing to the lung in vivo as well. So then continuing to build kind of on our arsenal of model systems, another really smart grad student, Matt Pajinski, joined the lab, and he's now actually an emergency doc in Kingston at Queen's. And he went away to a conference, and he had been working with the lung condition media model, and he liked it, but he was wanting to kind of expand it. And he went away to a conference, and he talked to a guy from the NIH in the US who had this 3D ex vivo pulmonary metastasis assay, or an assay called the PUMA. And what this assay involved is that you deliver breast cancer cells to the lungs of mice, and you can do this by injecting through the tail vein. It delivers them right to the lung. You wait about 15 minutes, and then you euthanize the mice, and you gravity perfuse them with a gelatin, which like fills the lungs up so they're nice and 3D. And then you put them on some gel film, give them some media, and you can grow them for 21 days. So Matt came back and he said, oh, came back from this conference. He said, I really want to try this. And I was like, there's no way that this is. And so the idea is actually the tumor cells will continue to grow in the lungs and form metastases, and you can study them. And I said, there's no way this is going to work. But Matt ignored me, like every good grad student should do, and went ahead and did the experiment. And sure enough, he started out just by looking at, just without injecting the cancer cells, just to look at the structure of the lung, because I told him I thought the lungs were going to die and not maintain their structure. And you can see they maintain really nice, healthy tissue structure. This is an H&E stain. This is a Mason's trichrome stain, which stains things like collagen and connective tissue. And so it does lose a little bit of connective tissue over the 21 days, but it's still pretty good. And then when you inject the cells in, then you can see that the cells go from the single cell to the micrometastasis to the macrometastasis stage. And then Matt sorted the cells into these ALDH high CD44 positive cells, and their non-stem cell-like counterparts. And again, he saw what we might have expected based on our other data, is that they grow much more rapidly than their non-stem cell counterparts. And you can just see the progression. The cells are labeled with a red fluorescent protein. And then if we think again back to that inefficiency, and that the key points are the ability to initiate the growth from a single cell to a micrometastasis and then maintain that growth into a macrometastasis, Matt saw that the ALDH high CD44 positive cells were the only population that could continue to make that progression, and the other ones kind of got stuck at that initial stage. So then we really wanted to know, our next question was really, can these lung-derived secreted factors, again, thinking back to the conditioned media where we're looking at soluble factors in the media, can these factors interact with these stem-like breast cancer cells in order to promote metastasis? And so we did a protein array on the lung conditioned media, and I'm not going to go into detail, but we did see about all of them, but we did see a number of genes related to mastagase genes, so soluble factors that would bind to those genes that were identified in the cancer cells or bind to the proteins that those genes made. There's a number of other potential metastasis mediators, but of great interest to us because of our interest in these ALDH high CD44 positive cells was that there was 5 CD44 interacting proteins. So that included osteopontin, which you might remember is a canonical ligand of CD44, but also some non-canonical factors, including a growth factor called fibroblast growth factor 2 or FGF2, as well as a group of proteins called the selectins. And all of these have a link in the literature to metastasis and bind to CD44. And so I'm just going to briefly tell you about, we are pursuing some studies on FGF2, and this is one of my postdocs, Dr. Vasu Bhatt, who actually did his PhD in Winnipeg and before he came to my lab. And so FGF2 is a growth factor and a signaling protein. Its canonical receptor is fibroblast growth factor receptor, FGFR, but it also binds to CD44, and it's involved in a number of processes related to stem cells and cancer, including development, angiogenesis, and metastasis. And when we used the PUMA model to look at FGF2, we used a blocking antibody, which is the triangles, and we showed that if we blocked FGF2 in the PUMA model using a blocking antibody, that we could reduce the formation of these metastatic lesions. And then Vasu's also doing a number of in vitro studies, and he found that if you treat with just parental whole population breast cancer cells with recombinant protein, either mouse recombinant FGF2 or human, then you can increase the proportion of these stem-like cells. So there's definitely a feedback loop going on between the seed and the soil. And so we have a number of studies related to how the lung microenvironment supports these stem-like cells, both completed and ongoing. So we know that these cells preferentially grow, migrate, and metastasize in response to the lung microenvironment. And this is because the lung is an important source of factors that can mediate breast cancer metastatic behavior. Potentially through these 5 CD44 interacting proteins, but probably other ones as well. And we're starting to understand that FGF2 promotes the stem-like phenotype, and we actually believe that it does so by talking to a transcription factor in the cancer cells called DAC2. And so I just want to, my time, wrap up in the last few minutes, just telling you, giving a little bit of a snapshot of where our current work is focused on. So one of the current questions we're working on right now is whether the lung microenvironment can be therapeutically targeted to prevent and or treat lung metastasis breast cancer. So I'm really focused on even our very fundamental science, trying to translate it to clinical work. So that is really our focus here. So the idea is that if you directly target the cancer cell, sometimes the cancer cell is so genomically unstable, it can just develop resistance. So direct targeting of the metastatic microenvironment is attractive because it takes advantage of the more relative genomic phenotypic stability of host cells, such as the lung cells. And then if you're able to deliver it directly to the lung, then potentially systemic toxicities or side effects of things like chemotherapy could be avoidable because you're actually just hitting the lungs. And so our idea was actually to use an inhaled approach, which of course has been used for many years to treat respiratory diseases like asthma. But this approach has remained underexplored for treating metastasis. And this is, Dr. Majumdar mentioned that we got a US Department of Defense grant, which was called a breakthrough grant. They only fund high-risk research, and they really like this idea. So we're collaborating with the Drug Development and Innovation Center at the University of Alberta and Dr. Raymar Lobenberg there. And he is making us some inhalable versions of these drugs that are targeting the factors that we found in the lung condition media, including osteopotin and FGF2, which we're using an inhalable antibody-based approach, and then the selectins. There's a pan-selectin small molecule inhibitor. And so that work is ongoing. We're just testing. So he's made the drugs already, and now we're testing them. And I have an amazing animal technician who is actually treating these mice with like a teeny tiny inhaler. And so we're excited to see that work. And then finally, the other really exciting thing, I think it's exciting, that we're working on right now is asking the question, or asking and answering the question, does the presence of an aggressive primary tumor actually further modify the lung microenvironment in preparation for metastasis? And if so, how? And the genesis of this question is that when we started presenting the lung condition media work at conferences, we almost always got the question. So you might remember we're deriving that lung condition media from normal mice. And so we often got the question, well, that's a normal mouse, that's the normal lung. What happens in a person with cancer who has a breast cancer? Is the lung the same or is it different? So the idea is that, and there was literature to support this idea, that the presence of a tumor, not just breast, but in our case, the presence of a breast tumor could actually somehow modify the lung to make it even more amenable to metastasis before the cancer cells ever arrived in the lung. And this is a really cool idea, first proposed by David Leiden in California, and it's called the idea of this pre-metastatic niche, so priming the secondary environment. And so a PhD student in my lab, Braden Medeiros, he started to look at that, and sure enough, when we compared lung condition media between non-tumor bearing mice and tumor bearing mice, particularly in animals with really aggressive subtypes, this one's called a triple negative, which is the poorest prognosis breast cancer, then the microenvironment was actually different. So there was some of the same factors, including all the CD44 ligands that we're investigating, but there was some other really interesting things. And so the question is how is the signaling getting from the primary tumor to the lung? And what Braden observed, and what's supported by other studies in other tumor types in the lung, is that we believe this message is actually being delivered through extracellular vesicles, which are tiny vesicles that bud off from the cell membrane. And originally they were just thought to be kind of junk or cellular debris, but actually they contain within them things like proteins, microRNAs, lipids, and then they get delivered to distant organs or other cell types, and then they can release their cargo, and that cargo can modify the microenvironment. And so other studies in other tumor types have shown these extracellular vesicles can affect matrix remodeling, inflammation, angiogenesis, breast cancer growth, and migration. And so ongoing work in the lab by two newer students, Irvi and David, we're looking at the influence of these EVs specifically on lung stromal cells. We're doing proteomics and functional studies on fibroblasts and endothelial cells, looking at whether we can actually target these cells therapeutically so they're not ever released from the tumor in the first place. So this is a prevention measure in terms of metastasis prevention. And then we're also exploring whether these can be biomarkers for lung metastasis in patients. And this work has a lot of similarities to Dr. Majumder's work because there's many of the markers are microRNAs. All right, so to summarize and kind of hopefully bring the ideas all together, from a seed perspective, we now know that these stem-like breast cancer cells display a higher propensity to spread to the lung. Bringing it into the soil, we believe this is because they're interacting through CD44 receptor ligand interactions in order to become able to metastasize and grow in the lung better. And this we think is through secreted factors, but we're starting to understand there's some matrix factors as well, and that these breast cancer-derived extracellular vesicles are priming the lung microenvironment in preparation for metastasis. And then I like a few years ago, I was asked to kind of give a broader kind of career talk, and this talk today was kind of a basis of that, but this is kind of where we've come from. So those of you who are interested in having your own lab at some point, we started out by identifying these aggressive cells. We didn't find any models that we liked, so we made our own models and tested them and validated them. Then we started using them to gain an understanding of the interaction between cells and help us answer our scientific questions. Now we're starting to identify potential therapeutic targets, and our goal, again, is to move this into translation to benefit patients either as biomarkers or therapies. So with that, I like to acknowledge, obviously, I've done so all the way through, but I have just an amazing team of students and postdocs and collaborators, as well as funders. And of course, our motivation of the breast cancer patients, we wouldn't be doing this work without them. And so with that, I will stop and we'll still have a few minutes for questions.
Thank you very much, Dr. Allan. That's very, very interesting. Yeah, I see some people are clapping. What I'm going to do now is open it up to questions. If you have a question, please raise your hand through the reactions or put your video on and raise your hand, and then I will ask each person to ask a question so we don't have people asking all at the same time. So it looks like Francis has raised his hand. So go ahead, Francis.
Wow, that's an interesting topic. I want to ask, how do genetic and epigenetic alterations within cancer cells impact their ability to interact with and manipulate the microenvironment to support metastatic progression?
Yeah, so that is a really great question. So in terms, I'll start with the genetic aspect. So you're probably aware there is a small genetic component to breast cancer, but only about 15 to 20% of breast cancer are caused by an inherited mutation. So BRCA1, the BRCA1 is a great example, but there are many others. And I'm not a genetics expert, but my understanding is the genetics aspect is usually related mainly to the initiation of cancer, so not necessarily to the metastasis. The initiation, so these patients get cancers much earlier and they tend to be more aggressive. The metastases are usually related to acquired mutations or somatic mutations that come along the way as the cancer cell kind of evolves and survives. The epigenetic question is a really great question because you probably know that, and not just in cancer, but many other diseases or situations, the epigenetics are dynamic. And so they basically, the goal is like they, unwrap the DNA or wrap up the DNA so that genes are accessible or more accessible or less accessible. And that epigenetics is often turned on or off by signals in the microenvironment. So again, we've done a little bit of this work in prostate cancer where we see a really big role for the epigenome. We haven't applied that to breast cancer, but I would bet money that it's definitely involved in the breast cancer microenvironment as well.
Okay, thank you very much. Hopefully that answered your question, Francis. You can put your hand down. Francis is probably getting digitally tired. Anyone else have a question? Masumi always has questions.
Mm-hmm.
Go ahead.
Such a nice talk, going from, literally from bench and really trying to reach up to the patient. There are so many aspects. I don't want to hold up the whole question answer period, but I was just curious to know when you are trying to grow, develop an ex vivo model like the lung, what was the greatest hurdle you had to face, other than by concept that it's not within the entire body, but outside? What was the hurdle with respect to growing and managing them you had to face when you are trying to establish the model?
Yeah, so I think I would say overall, it was kind of finding the right media configuration, so putting the right growth factors in, and supplements and things like that. So you can imagine primary cells are a lot fussier than immortalized cells. So we've had to play around a lot with that. And then interestingly, we saw some differences in the different organs. And so you might have noticed that I didn't show a lot of data related to the brain condition media. And so the brain has a lot of lipids in it. And the lipids, just when they went into the media, it was just super messy. And I have a lot of neuroscience colleagues, and they're like, of course, brain is messy. So it was sort of I guess, overall accommodating organs that normally grow in this really rich microenvironment, and kind of trying to convince them that they want to grow in this very simplified sort of culture system, and playing around with how we can support them without changing their biology too much.
Yeah. So many organs you dealt with, so understandably not every tissue is similar. I can see my lab there raising hand, so I'll leave it to them to ask more questions. I can see my grad student, my honor student sitting there. Go ahead.
We're all
together. Go ahead. Go ahead, Reed.
I'm Brady. I'll ask the question from
okay.
So more of a general question. You mentioned that you had some unexpected results when exposing these cancer stem cells to different conditioned medias. How did you sort of approach those unexpected results? Did you question them at first? How much did you dig in to make sure you got the proper results? And then how did you change your approach moving forward?
Yeah, no, that's a really great question. And so, so I think the situation, or the experiment that I kind of specifically mentioned was, again, we wanted to look at these stem-like cancer cells and see if they had a preference for any of these organ microenvironments. And we went into it as you do as a biologist, as you're trained, with a hypothesis. And again, we hypothesized that of the 5 or 6 or the 5 conditioned medias that we looked at, they were definitely we were convinced they were going to like the bone best, because there was lots of evidence of a rich stem cell niche in bone. And then when it showed up as lung, then my first thing was are you sure? So I always say to my students that it's way easier to explain human error than it is to explain biology. So I always, whenever we get an unexpected result, we always go back and do the experiment 3 more times on top of the original 3 and make sure every single little technical thing is taken care of. And then if we get this result, then we're happy that it's sort of a legitimate result, then we sort of go, oh, well, maybe this is interesting to pursue it. And then historically, a lot of the most interesting and impactful findings in science, in particularly in cancer biology, have been because of serendipitous results of things that didn't work. Like my favorite story is Judah Folkman, who developed the first anti-angiogenic drug therapy. The reason that drug was developed is because a student in his lab, I think, went away for the weekend, left his cells, the cells overgrew, they became contaminated with a fungus or a bacteria. And then when he came back, he was almost ready to throw them in the garbage. And he looked and he saw that none of them were forming blood vessels, whereas the other ones were. And so he extracted whatever was growing that he was going to throw away. And then that led to this kind of finding of that. So never, control for all your technical variables, and then stand up to your PI to say, I know I've done all my hard work and it's a legit finding, and can I, kind of pursue it, because sometimes the most unexpected things are also the most interesting and important.
And never throw anything away. We've had a few we've had a few speakers say that. I remember Dr. Wendy Untriner said that last year, because she's now working on something that someone did years and years and years ago. So well, it's 3:31, so I don't see any other hands. So maybe we will give Dr. Allan another thank you very much for taking your time and energy and willing to come and talk to us today. It's very nice to have someone of your stature at our little university. So thank you.
Oh,
Thank you
my
very
pleasure.
much.
Thank you. Right. Have a great weekend, everybody.
Yes,
Thank
have
you.
a great weekend,
Thank you.
everybody, and we'll hopefully see you in person mostly next week on Friday.